Welcome everyone to the oncolytic virus session at this year's SACS event. Unfortunately, virtual once again, no longer the day preceding ASCO, but hopefully we'll be seeing each other in person at ASCO or at Bio or at SITC or any number of other events, barring the rise of monkeypox, if you saw the stat news this morning. So could be the decade of viral epidemics. Anyway, I'm joined by a stellar cast of people with experience both in oncolytic viruses and immuno-oncology and broadly in oncology from both their current purview at these 4 biotech companies as well as their past history. I will let each of them introduce after I just do a brief introduction to the space for some of those who may be less familiar with oncolytic virus, and then we'll get into hopefully a good, rigorous discussion, debate, dialogue. So I'm just showing this very briefly just to frame the fact that, for vaccines, oncolytic viruses and other things, we've been thinking about it for a long, long time. And, this was a presentation from Sanofi Pasteur in 2006. And basically many of the issues then that applied to either vaccines or oncolytic viruses are still applying today. This question of what's the best platform if you're delivering or hoping to get a vaccination approach. With a directed antigen, what antigen or antigen should you choose, or are you relying on in situ vaccination? What immunostimulatory agents do you put in as transgenes, et cetera? So, these same issues now, 15 plus years later, are still being discussed, and we'll be debating them and talking about them on today's session. One thing for sure, that's changed, and I borrow from the famous film, The Graduate, one word plastics, one word checkpoints. They really kind of changed and enabled the world that we're in, kind of converting what I call this backwater of what had been tumor immunology now into the exciting and very hot space of immuno-oncology, with a variety of effects that are important for clinical benefit, for conducting clinical trials, et cetera, both for oncolytic viruses or OVs, as I may refer to them, in cancer vaccines. We don't have a big selection of approved oncolytic viruses worldwide, and in the US, we really only have T-VEC, Mlygic, and we'll talk a little about the overhang, perhaps, that Mlygic represents for the field. One thing for sure we're going to touch on is the fact that there are many, many different families of viruses that various people over the years and currently are trying to exploit as oncolytic viruses. In fact, we'll talk about the misnomer of the name oncolytic virus, but many various families that people are working with in order to try and accomplish ultimately the same goal of getting perhaps tumor cell killing, but certainly, kind of igniting that fire within the TME and kind of bridging that innate adaptive landscape. And we'll talk about some of these issues as well. The real elephant in the room is this issue of delivery, and that may be part of the overhang with Mlygic is, biopharma generally doesn't meet the checkbox for kind of this issue that's seen as an impediment of local delivery or intratumoral delivery. And, a large proportion of viruses that are in development are in fact still being delivered in some local way. And so that is an important issue to discuss because there are pros and cons to that idea. From a much more practical standpoint, when you look at oncolytic viruses or cancer vaccines or some of these other modalities that have had some troubled history, you see it's still a reasonable amount of activity when you look at the pipeline. But in fact, when you look at analyst consensus forecast sales, not a big impact for OVs or for cancer vaccines. If you look at venture funding, for example, not a lot of investment going into those spaces. They're being sucked up by, all sorts of flavors of antibodies and adoptive cell therapies. I don't have a pharma partnering slide in here. I do have one, but maybe I'll stick it in later for the deck for anyone who wants to access this and ask me. So this is the framing for the discussion that we will have with some of the issues that everyone in the field that will be facing. And now I'd like to turn it over to each of the panelists to allow them to introduce themselves and their platform. And we will go in the alphabetical first name order that was reflected on the first slide, which is Angelica, first you from Lokon. Thank you.
Yes, thank you, and hello everybody out there. So I am Angelica Loskog, a researcher from Sweden that worked with oncolytic viruses for about 20 years. So I did trials already back in 2006, but then with replication deficient viruses. So today I am the CEO of Lokon Pharma, which is a Swedish biotech company that develops the load adenovirus platform. And our lead candidate encodes 2 human immunostimulatory genes, the CD40 ligand and 4-1BB ligand, and load 703 is currently being developed in 4 different clinical trials. So our major focus is on pancreatic cancer, and we are now aiming for phase 3. So the next slide, please. So what is Lokon's concept, and what makes us then maybe a little bit different from others? And I would say that it is because our transgenes are expressed both in the tumor and in the stroma. So virus replication can only occur in the tumor when a tumor cell is infected. But if the virus is infecting a tumor or stroma cell nearby in the tumor, the stroma will help to produce these transgenes. And with that, we hope to inflame the tumor lesions. So next slide, please. This is our clinical program. As I said, pancreatic cancer is our major focus, and we are treating these patients in Lokon 001 and in Lokon 002. We are combining it with chemotherapy, but also with the addition of atezolizumab and anti-PD-L1 antibody. We also have a trial for malignant melanoma where we combine our virus with atezolizumab, but without any chemotherapy. And then Roche has initiated a trial trying to combine our virus with their atezolizumab in colorectal cancer. So there's where we stand now, and I will leave the word to the next panelist.
Great, thank you, Angelica. Joe, you're up.
Thanks, Jeff, and I'm very excited to be here with this esteemed panel. My name is Joe Kaufmann, and I'm Executive Vice President for Oncology at Codagenix. My background is in molecular medicine and oncolytic virus engineering, and I've spent the last 8 years or so in industry translational R&D at the intersect of infectious disease and immuno-oncology. Codagenix is a synthetic virology company harnessing the redundancy in the genetic code to create safe and potent immune stimulatory viruses that we are applying in 2 different disease areas. Historically, we have used these viruses in the form of live-attenuated viral vaccines for infectious disease prophylaxis. And in this area, we've established a robust pipeline that includes 3 clinical stage programs, most prominently Cobiliv, a live-attenuated intranasal COVID vaccine that is entering phase 2-3 trials sponsored by the WHO. More relevant for the discussion today is our new immuno-oncology vertical, where we're applying these viruses in the form of oncolytic viruses to unlock the potential of barotherapy of cancer. I think that's all what we're here for to discuss today, how we can do that better. On the next slide, we illustrate at a top level some of the capacity of our proprietary technology that is the foundation of the company. This is a synthetic viral engineering platform, also known as SAFE. The foundational biology behind this platform is something that is unique across all viral species, and that is that they have optimized their genetic information and their codon usage for optimal translation and replication in their respective host cell. Now, our machine learning enabled algorithm reverses this process in selected genes of a particular viral species by introducing hundreds of silent mutations into these genes. These mutations do not alter the amino acid sequence, and therefore all the immune stimulatory components that the virus naturally possesses are retained, and therefore we believe that these viruses can contribute to holistic modulation of the tumor microenvironment when we think about this in the context of cancer. What these mutations, however, do is that they alter the codon usage of the virus to introduce less favored or codons or codon pairs. This means slowed down translation of those codon-modified genes. And in the context of a viral replication cycle, this means slowed replication in cells that are otherwise immune sensing competent, for the purpose of this discussion, healthy cells. However, this attenuated phenotype is less prominent in tumor cells that have defects in a variety of signaling pathways. Therefore, this codon modification adds a layer of tumor cell specificity. Another key feature of this platform is that it is species agnostic, and therefore we are approaching discovery in a patient and histology first manner by tackling an unsolved question in the field that we may or may not get to later, that revolves around what is the right viral species for tumor type of interest, and our screening platform aims to address that. In parallel to that, we are progressing our lead asset, Oncolytic, toward the clinic. This is an influenza-based virus called Oncolytic, which we're developing in the context of immunotherapy of breast cancer. We're completing our IND-enabling studies and are hoping to enter first-in-human clinical trials at the change of the year. We are a spin-out of Stony Brook University, headquartered in Farmingdale, New York, on Long Island. However, we are currently expanding our virotherapy team to Cambridge, Massachusetts, and we look forward to start up oncology operations in Cambridge in the summer. And I'll stop here, and I look forward to the discussion.
Thank you, Jo. All right, let's transition over to Lubor.
Hi, my name is Lubor Gaal. I'm the CFO for Targovax, and I'm very pleased to be also speaking on this panel. Thank you very much for giving us the opportunity to be on this panel. Maybe we skip to the next slide. This is a forward-looking statement because we are a publicly listed company in Oslo, Norway. And we have been in the oncolytic virus space probably for over 10 years. The original technology came from Finland, same place where Valo, the next company, is also from. Basically, our lead asset is Oncos 102. It's an genetically modified adenovirus, which we have tested in different indications or different tumor types. And most recently, we have shown very strong clinical efficacy in PD-1 refractory melanoma. With a, I would say, very competitive, not class-leading 35% overall response rate. What really gets us excited about is actually that we have the matching biomarker data, which I will show you very briefly in this presentation. We also have a KRAS vaccine and a circular RNA, but I will focus mostly on talking about the oncolytic virus program for this panel discussion. Next slide. Here you can see a response from this melanoma trial that I mentioned earlier. As you can see, patients have responded, or the people who respond, the patients who respond very quickly to our oncolytic virus therapy. Overall, the durations are durable. Keep in mind that dosing has stopped after week 9, so there's no more injections in those patients. So very favorable, very effective clinical responses. But what gets us equally excited is the next slide, where we can really match the responses from responders versus non-responders. So here you can see responders in blue versus non-responders in red, and you can see a clear separation at week 9, where we really see strong activation of the immune system. So, of course, we have very much detailed genomic analysis, and we can equally specify, of course, which are these targets. Some of these targets are very well known in the immune therapy space, some of which are novel. But what we're trying to highlight here is that actually this virus is not just, of course, affected maybe because of some oncolytic or oncolysis, but very much by activating the immune system and really triggering a strong immune response in those patients and showing really good efficacy. And I really look forward to speaking more about this in the panel. I think this is my last slide.
Great. Thank you, Lubor. All right. Oh, you had that one.
Yeah, it's just the match that we also had a clinical trial in mesothelioma where it's combined with, so the other one was in combination with PD-1 and PD-1 refractory patient. This one is in combination with chemotherapy. And as you can see, we have the longest survival rate, median survival data of all the studies done in this study, even longer than IPI and Nivo combination, the same patient population. So not just works with PD-1 checkpoint inhibitor, it can also work with chemotherapy. So with that, I turn over to the next speaker.
Great. Thank you. All right. Paul.
Yeah, thanks very much, Jeff. Paul Higham, the CEO of Valo Therapeutics. I've been in immuno-oncology since 2008, working to try to develop new therapies for oncology since 2001. and in the industry in total for 37 years. I'm really excited about immuno-oncology. I've always been interested in oncolytic viruses. So when the chance came to join Valo in August of 2020, I jumped at it. So Valo is a spin out from the University of Helsinki, spun out in 2016, and the founders of the company, some of them were involved in the base technology that's being used in Targovax now. So there's some commonality there with Lubor's company. So we have, I think, something a little bit different in terms of oncolytic virus technology. We've got a patent that allows us to attach tumor-specific peptides to the outside of oncolytic viruses, to use essentially the oncolytic virus as a delivery vehicle for tumor-specific peptides. What we do is we identify the peptides that are relevant to the particular tumor, and then positively charge them by adding a polylysine tail. And with the virus being negatively charged on its surface, we create an electrostatic complex. So the virus is essentially then coated with the relevant peptides. That's then delivered in by intratumoral injection to the tumor. And what it does is obviously drive a very specific T-cell response against the antigens that we've delivered. The oncolytic virus is conditionally replicating, so only replicates in tumor tissue. And the viruses that we're using, again, which we have patents for, have got particular transgenes to try and supercharge the immune system. So you get like the double whammy of the oncolytic effect of the virus antigen spreading from the tumor cells being destroyed, but in addition, these very specific targets of the peptides we've attached to stimulate additional T-cell response against the tumors. We're going into a phase 1 study in a matter of weeks in 3 indications: melanoma, triple-negative breast, and non-small cell lung cancer. And we're going in combination with pembrolizumab's checkpoint inhibitor, so charging up the immune response in weeks before then delivering the checkpoint inhibitor. We expect that to be a very synergistic combination. I think the difference in our platform is the extreme flexibility we have, because it's so easy to attach peptides. We can very easily switch in and switch out the relevant antigens depending on the tumor, or even, we hope in future, the specific patient that we're treating. So we'd analyze the patient's tumor, identify the targets, the peptide sequences most relevant for that particular tumor, and then be able to deliver that back to the patient in an individualized therapy. And because peptide manufacture is so inexpensive, we can do this, we think, at a fraction of the cost it would cost for some of the other immunotherapy approaches, adoptive cell therapy, CAR T, etc. In addition to this lead platform, which, as I say, is going into phase 1 any second now, if we go to the next slide, we have another of other platforms, a similar platform to cover envelope viruses, where we think we could add an even greater payload of antigen to the outside of those. We're in preclinical studies with that. We're also looking to use it in infectious diseases, where we choose infectious disease agent-specific peptides to create a T-cell response. We're working on a pan-coronavirus vaccine preclinically. And we also have a patent related to the use of the TB vaccine, microbacterium also being negatively charged on the surface, same concept, and positively charged peptides that are relevant for particular variants of TB. And we think we could create better TB vaccines than are currently available in the world. So that's the platforms behind the lead, which is Peptigrad. I'm really looking forward to the discussion.
Great, thank you, everyone. I think we've set everyone up to kind of appreciate the perspectives for each of you and where you're coming from, both from your own individual backgrounds and from the companies. So, you know, we have a variety of talking points, whether we get through all of them or not, and we won't go in strict order, because I think we'll end up touching on a number of them, you know, at the same time as this discussion evolves. Let's start with the top one, because this is something that probably everyone faces, regardless of which virus, which platform, what your engineering is, which indications you're pursuing, which is oncolytic virus, intratumoral virus, you know, kind of a double whammy there. So this baggage of history is something I think that everyone has to face right up front, whether with prospective partners, investors, et cetera. So who wants to leap into the fray first on this topic, and how we're kind of trying to move past it, and what we think we can continue to do to improve the perception and understanding? I will go to you, Angelica, just because you have the longest 20-year history. So by virtue of that, we'll start with you.
Yeah, well, this is actually a very difficult question to address, I would say, but I think that it's important that we all understand that this is a field that has been running for quite a while, that has faced different obstacles during the way, and it's also, as I usually say, two fields that has been merging, the oncolytic virus field and the immunostimulatory gene therapy field. And I think that we need to somehow define what we are, and I think that might actually be different for different OV companies. And some of us might focus on getting rapid and good oncolytic effect in the tumor, which is one thing, and others might focus on having the immunotherapy effect, and some hope to have both, and so on. And a little bit they contradict each other, because if you have an oncolytic virus that also provides immunostimulatory genes, if you have too much oncolysis or too fast oncolysis, you will limit and reduce how much transients you will have expressed. So it is a double-edged sword all the time in a way, and I think we really need to define for ourselves what is the focus of our company and what is the goal for our product, and try to lift that when you go out there and talk to investors.
Thanks, Angelica.
Lubor,
look like you're ready.
Yeah, I take, maybe a different view is that, why should oncolytic virus be any different than any other platform technology? I mean, like we all know that in our industry, there's always a lot of hype about something that is new, and maybe people are more simplistic about it and hope that it leads to very quick results, and then realize, wow, the road is a little more rocky. It happened to antibodies, happened to checkpoint inhibitors, happened to a lot of other things. And I think the same happened in oncolytic virus. I mean, and Jake is totally right. The field has completely evolved, and things have changed, because we have learned things that we thought would be really easy turned out not to work so well or so. And I think this what happens in our industry all the time, and I think it will happen in the future with other novel platforms again or so. And she's right. I mean, are we looking at in the old days? I think everybody was looking at an oncolytic virus from its oncolytic potency. Can it really eat up the tumor by itself? I think we have now all evolved and thinking that that's probably not the road to success. It's more a combination of this, and this is what our does. Oncos 1 or 2. I mean, yes, it does have, of course, oncolytic potency, and it does kill tumor cells, but that will not kill the tumor. So you only can really have a strong anti-tumor efficacy if you also activate the immune system, and that's what, and Jay was saying, it's very important what you put in your virus, that's important. And she's right. I mean, you have to play now with different dimensions and parameters versus when you want to express a certain protein that has other functions. But I think that's where there's a lot of questions. It's a multidimensional, but what is nice about it, and what we saw maybe in our data, is that you have different dimensions of efficacy. You have the direct tumor killing versus the immune activation versus the, spilling out maybe oncolytic peptides, neoantigens, or can activate the immune system. So there's many different, so it's a very nice thing if you like multidimensional problems. Of course, it's not an easy solution.
Thanks, Lubor.
And maybe adding to the theme of let's take the learnings from the experience and build on that, not only on the scientific side. I think there are other things that we can take away that the last decade or more of oncolytic virus clinical development has taught us. I think we generally can conclude there's an exceptional safety and tolerability profile of this modality, which opens up interesting clinical avenues when you think about how to pair these agents with standard of care, whether you think about replacement or early stage treatments. I think we can go into the clinical development path for a panel on its own, but I think there are learnings in there that we can build upon, knowing that we need to improve on the efficacy side, and we all have our own way of thinking about how we can tackle a component of that.
Thanks. And then maybe to you, Paul, because again, I think this idea of the pleiotropism and the straddling between the tumoricidal versus the immunostimulatory aspects, and clearly, there is an overlap in many cases, directly or indirectly, between OVs and vaccines.
Yeah, I mean, I guess at Valo we're trying to take the best of both of those worlds really and combine them. And I to Lubor's point, I think it's always the case that these, a new field, if it's going to work, takes some time to evolve. And I think there's a dramatic difference in what we're doing now, compared to what was being developed 8 years ago. So I think we can be very optimistic about that. And a little to Joe's point as well. I think by the way, we should probably rename the whole field. We should stop calling it oncolytic viruses, right? Because we're really immunostimulatory platforms of various kinds. And that may help us, may just be communication, marketing, but actually I think it's real. I think that change has been a real change over the last 8 years. And to Joe's point, I think we've got an amazing flexibility that we can bring to the party with our platforms, particularly when you think about, what's the overall ideal oncology therapy going to look like in the future. You may be combining checkpoint inhibitors. You've got immunostimulatory agents such as the ones that we're talking about, trying to drive, a strong specific T cell response. And that's going to become incredibly complicated and expensive to deliver and actually, for healthcare systems to pay for. And I think we've got the opportunity also to deliver kind of real world T cell stimulatory systems that healthcare systems can afford when you compare it to the complexity and cost of CAR T and adoptive T cell therapy approaches. I think there are distinct advantages. We need to deliver the clinical data to prove that, but I think we're on a good track to do so. Yeah,
and it's not without intention when I often say OVs versus oncolytic virus. It's a way to kind of back away without creating a new acronym, which maybe we'll get to. But so everyone, either in their talks, et cetera, has kind of alluded to kind of the role, at least in clinical development, and probably in terms of clinical benefit, mostly of use relative to combined with, checkpoint inhibitors. And certainly, aside from advances in the understanding of what these viruses can do over the past decade, it certainly, we cannot deny, been incredibly facilitated by the approval of checkpoint inhibitors, or CPIs as I use here. So any kind of comments about, how you're working with these or relative to these? Because certainly, it applies not just to OVs, it applies also to CAR Ts and others. Sometimes they are engineering in, whether it's OVs or others, engineering in the checkpoint, as opposed to, needing to partner with an exogenously applied one that introduces different kind of clinical development, regulatory challenges, et cetera. But I'd just like to hear briefly some of your thoughts about kind of the role, your platforms relative to development with checkpoints. And that also has partnering aspects too. And since this is an investor and partnering forum, I think we want to kind of bring that up as well.
If I could go on that, maybe Jeff first. It's I think actually, as you say, checkpoint inhibitors have facilitated, I think, an enhanced development possibility for our platforms, particularly from our perspective when we were designing our phase 1 study, and we're going with pembrolizumab, which is by far the most successful checkpoint inhibitor and has the broadest set of indications. It kind of helped us come up the ladder of stage of use for our therapy. So because we could link in with that, we weren't getting pushed right back to the late stage patients where we knew their immune systems would be damaged and it would be difficult to show an effect. And because of that, we're more optimistic. So I think there's been a fantastic facilitatory effect of checkpoint inhibition. Although, having said that, my comment earlier, I think we're probably going to need to look at more than one checkpoint mechanism to really get to a very high response rate. Checkpoint inhibitors typically 30 to 40% now. Can we enhance that with our platforms to 60, 70, 80%? But to really crack it, I think we've got probably to look at another checkpoint axis to add into that. And again, the cost aspects, as I mentioned before, come into it then, you're into triple therapy potentially. But no, I think it's been a great benefit. And actually, I think it would have been difficult to imagine developing a study where we had a lot of optimism without checkpoint inhibition also being there.
Anyone else?
Yeah, I mean, I think as always, it's a decision where you have to take multiple components into consideration. Like when I was at BMS, I know we always said science has to lead the way. So of course, clearly this combination has to make scientific sense. And, in the data that I showed you, and when we can go more in details, we can really show the synergy between our virus and checkpoint inhibition, and then that it makes sense to really combine it with checkpoint inhibition to get a better response. And that's the other consideration. I mean, as we know, we're in a very competitive field. I mean, we're in melanoma. Melanoma is very competitive. Where oncolytic virus is not the only modality that's tested. So you have to make sure that your response rate is competitive with other modalities. So you increase your chance, of course, in combination. I have no doubt that the virus itself would be effective, but it probably wouldn't reach the efficacy level that you need to be competitive with other approaches. And as Paul was saying, one thing that is a growing problem in our industry, of course, is people who are refractory to PD-1 treatment. Even though PD-1, of course, is a great treatment, is a great drug, and lots of patients benefit from it, there's many patients who either can't take it or don't respond after a while. And that number, of course, will increase with increased use of PD-1. And I think it's going to be a huge unmet need that we need to solve. And so, I mean, people use it before and make cold tumor hot again, or so whatever they talk or say about it. So oncolytic virus may be an ideal vehicle, to restart the immune system and make PD-1 effective again, either in tumors where they don't work or where they stop working. And so we have lots of data that makes it the logic choice. But Paul is right. I mean, one needs to probably look beyond PD-1 and not just look at that.
Yeah, and I would just say, because I know we're often, including myself, very commonly using the colloquial expression like turning cold tumors hot. But if you're in potential collaboration or partnering discussions with Emmett Schmidt at Merck, do not use that phraseology. Anyone else want to comment about relative use or role of checkpoints or?
Yeah, well, because I think it's a very, it's the perfect marriage, almost, as you said here. You have these fantastic viruses that do something very well, and that is attract T cells. So, but something that we do need to solve is for the virus to reach more tumors. And if we are doing intratumoral injections, we will have a great inflammation there, and we will have the vaccination effect that can give an abscopal effect. But there is another part that we sometimes forget, and that is the capacity of the virus by itself to attract T cells to the area where it functions. And the vaccination effect is good, but it would be even better if you could inflame all lesions. So delivery methods for viruses where we avoid neutralizing antibodies and so on, that is something that we need to work with a little bit more in our field, I think.
Great. I'll pick up on that in a second. Joe, did you have something you wanted?
Yeah, I guess I wanted to, first of all, I agree with all the scientific rationale for this combination where there's, complementary mechanisms of action, at least for the majority of checkpoint inhibitors that are more about sustaining T cell function. And I would say that goes beyond PD-1, PD-L1, includes now LAG-3, but also others that are earlier stage in development. But to come back to the point of, should it be a transgene encoded in a virus, or should it be an add-on as a whether part of standard of care or not? I think there are benefits to the non-transgene approach in my mind, in the sense of the biology of systemic T cell releasing of the brake, so to speak, the clinical development aspects about add-on or replacement of components of standard of care, and clearly you wouldn't replace the checkpoint component of that. But there are also questions around sequencing and other things related to maximizing the mechanism of action of these Viro therapeutics, trying not to say oncolytic virus. Where also you have a lot more flexibility to really assess that systematically by using them as independent agents, and in some ways developing combination regimens rather than encoding the checkpoint inhibitor, whether in form of a single chain of B or VHH or whichever format you have coding capacity for, to do that within the virus and only express that locally And that comes back to the kinetic question Angelica already mentioned around transgene expression efficiency versus the replication component of the virus.
And it also brings us back to the value of local activity versus systemic activity, and whether that's due to the virus being able to get not only the primary tumor, but various sites of metastases, whether those are macro or micro. I think is an important point that we can segue into. Paul, you were starting to make a comment, or?
Yeah, I was actually linked to that entirely, Jeff. So I think the question is, how confident can we be that we can create a vaccination effect at a particular tumor site where we're injecting our therapy that's going to create a systemic effect? So, I'm much more excited about a systemic immune system effect than systemic therapy. Right. So I think with viruses, it's always going to be a real challenge to try and get past the immune system and have something that's going to reach all parts of the body and still get there in a concentration that's really going to have an effect. So I think we really should be focusing on, can we use the one tumor site to generate such a powerful vaccination T-cell essentially effect, where the T-cells then with checkpoint inhibition in place as well can go to the other tumor sites and have an effect that's going to really affect the overall tumor burden. I think go ahead.
I was just going to say, let's tie some of these themes together then. We have this issue of the transgene and the local activity, systemic effects versus actual systemic therapies. There's the point I raised about kind of that checkbox that pharma or investors have about kind of IT delivery. Let's get very practical to the degree that you can talk about, like, what types of, I presume this is being raised with you in real world constantly, this issue, whether it's pharma partners or investors or other stakeholders. How are you kind of messaging it right now, and kind of what do you each think the bar really is? So if you're acting locally, whether you've given it locally or not, how much benefit do you have to show in order to show that you're actually having that systemic benefit? Is it the percentages that, you alluded to, Lubor, kind of above and beyond checkpoints? Are kind of investors or pharma looking for something even more, or who wants to kind of leap into the fray on this one?
If I could just very quickly, I'll hand over. I just say, I think overall response rate is obviously, a key measure that we're going to look at, and we do need to see an abscopal effect. We do need to see an effect on tumors distant to the injection site. Otherwise, we're not demonstrating a systemic immune system effect. We do need to be able to show that. And then I think actually, if we can show that, the idea of IT being a difficult barrier kind of goes away. Strong, powerful clinical data, objective response rate data will overcome any difficulties with injection in this type of difficult setting any day. So I think once we get to that point, the IT issue then starts to go away. I think it's that, in the therapies that we've seen so far, we haven't seen a strong enough systemic immune system effect to convince people that it's worth going through the hassle of IT.
So let me push back a little bit, and maybe that might be a segue to Lubor. I don't know. We'll see. Is that, we've certainly seen, not just with OVs, where you get a disconnect between ORR, right? Because you're talking about effect on the primary tumor versus what we're talking about here is an overall systemic effect to basically, at the end of the day, hopefully lead to increase in overall survival, which means it's not just the effect or the organ dysfunction caused by the local disease, but it's kind of the broader systemic disease that is the cancer. So
absolutely. Jeff, you're absolutely right. And in the end, I think always, it depends who you ask. You get a different answer. So like if you talk to the guy the payers, who represents insurance company, like you said, he said, probably as you said, I don't care what the ORR is. I need to see overall survival data. Does it really make a difference in the end? So I know if you ask a clinician, you get a different answer. If you ask a biologist, you get a different answer. So, I'm on board with Paul and saying, just giving a drug systemically doesn't, by the default, makes it better. Actually, to the opposite. I think we would all agree that putting the virus where it needs to be actually makes a lot of sense. So why would you go away from IT? Because you've got to put it where it needs to be. So on the other end, then you have the commercial guy who says, yeah, but blah, blah, I can't sell this. So as always, we're in a world where lots of people pull you in different direction, and you have to make decisions which ones is the most important. But I think in the end, the most important is we have to show that it works. And so we need to think about what is the best conditions, in what condition would our, let's say, immune activating therapy do its best job. And I think every company that has a different virus, that has different properties and probably may come to a different conclusion here. So, and that will be interesting to see, which path will get you to the finish line, so.
Yeah. Joe, Angelica?
Yeah, this is actually one of my favorite questions, because what about response rate and what about survival in immunotherapy? Because this is actually something that can be contradictory. You can have a good response rate, but poor overall survival, and vice versa. So the immune system works in a very different way, and you have to find out what is the purpose of your treatment. Is it to have the fast response rate, or is it to have the longer overall survival? Because if you have a virus with fast replication or that has an initial good immune response, you might get tumor reduction. But if you have another type of immune response growing over time, you may have a tumor control that leads you to a really long overall survival. And this is actually important, because in the end of the day, if you are a patient, you will choose the product that will make you live longer. But then you want to live a good life while you are doing it. So I do think we have to think more about this, and also how to evaluate overall survival, because if you look at median survival, that is something that also tells you something that happens rather quickly. But with immunotherapy, you have this tail of patients that survive longer, and then median survival will not address that. So you need to start to look at area under the curve and different ways of doing analysis of your survival curves.
Yeah, I'm glad you brought that up, and it was on my slide about the one-word plastics with the checkpoints, that the following the trials longer, following the patients, looking at that tail curve. It is quite conceivable that even without the availability of checkpoints for many of those first generation OV or cancer vaccines, that there certainly were patients that were probably responding. They just didn't get followed long enough. And there were probably only a few of them anyway, since there weren't checkpoints. So they were already facing a certain impediment. But Joe?
I think I agree with survival-based endpoints being maybe the more valuable output long-term, while that may be painful for small biotechs like us, who therefore have to invest a lot more money upfront to actually follow these patients for that long. We still have to realize that survival-based endpoints are typically the ones that are the established approvable endpoints, and that's, I think, all of our ultimate goal. So we do have to tackle that survival-based endpoint at some point, but maybe it begs mention of changes also in how we assess responses in general. There are evolving concepts about IT resist and other things that are addressing some of the issues related to intratumoral agents, whether that is virotherapy or other local stimulatory agents that are being given, that allow the choice of which lesions to inject, which we may not be needing to use one. It can be multiple injectable lesions, which can help us with this transition from a local immune stimulation that, by the way, qualitatively might be different than if you were to inject a virus systemically, to this more systemic therapeutic effect that we're all hoping for. So I think there's more interaction with regulators that are required to help us all get there and how to appropriately assess these modalities. But ultimately, coming back to where we started, the efficacy needs to be there systemically, and it needs to make an impact. And once we prove that, I completely agree with Paul. The discussions around equity and ease of administrations should wane.
Absolutely. Yeah. So I'm curious. So again, tying it into the kind of investor partnering angle clearly here, front and center for SAC's event. And I want to go back to something you said, Lubor, about, the competition is not just in kind. It's not just another, say, adeno-based virus. If you're adeno-based, it's not just another different family of OV. It's like a whole range of modalities that are potentially poised, antibodies, bispecifics, adoptive cell therapies of many flavors, small molecules, next-gen checkpoints, on and on and on. So let's assume you get past in discussions with partners or investors, you get past this bugaboo of the history, you get past the issue of kind of what the clinical trial can be like now that there are checkpoints and where you might be used relative to that. You get past the IT, if your IT is part of the value, aside from obviously, the data is the data, but is part of the story, the pleiotropism that you alluded to, Lubor, and we've all kind of touched on, this kind of straddling innate, adaptive, it's killing the tumor, it's recruiting or activating immune cells. Do you find that is an important selling point relative to this broader competitive world of many different types of modalities?
Yeah, I mean, like I said, I worked for Bristol Myers Squibb for 10 years, and I was looking on the other side, looking at lots of alternatives. And I always would say to my scientists, the patient doesn't care how the drug works; they only care that it does work. And so that's pretty much the same here. I mean, I don't think that any pharma company has high on the wish that I need to have an OV program in my portfolio. They just want a very effective drug treatment that help patients, and they will look across any modality. So I think we need to compare ourselves not in the space that you are in, the therapeutic area to focus. You have to look beyond just the OV space. You have to look beyond all the other modalities, and you have to be confident that you're going to stand out and that this approach is the best. And then it depends a little bit on your philosophy. I mean, so I know that in a lot of pharmaceutical companies, they love this one target, one effect kind of, they like those clean drugs or so. But when I worked at a biotech company 20 years ago with a very famous German tumor biologist, he said, if you only hit one target, a cancer cell will get over it. It will get around it. There's so much pleiotropism there, like that it has a way to get around it. So one single target bullet probably won't work with a cancer cell, So you have to hit the cancer cells in many different ways, and that what an OV could provide by activating the immune system. But it depends on what the head of research at that pharma company believes.
Yep.
In a way, it's multiple shots on goal by having multiple components of the cancer immunity cycle being impacted by these viruses. And as we said, different viruses have different qualities, and which of those might be more strongly affected by them in a particular cell environment with certain sensing pathways upregulated or not. So in a way, it actually helps addressing some of the breadth questions if you have multiple mechanisms of actions you can leverage and rationally design combinations and combination regimens with. So I agree that this is a valuable component.
Yeah,
I think go ahead, Paul.
Yeah, sorry, I was just going to say, I think having a complicated immune system means you shouldn't aim for a simple therapy in a way, right? So you want to press as many positive buttons on the immune system as you can to have the greatest effect. I think one of the challenges we face, a practical challenge in the clinical studies, is being able to demonstrate we're actually doing that. So the immunomonitoring part of the picture, when we go into clinical development, we're just putting together our immunomonitoring plan for a phase 1 study. And we're trying to cover absolutely everything, to see everything that we can see. And I think that actually communicating, assuming we get positive data, communicating that then to potential partners that may not be familiar with the complexity of the immune system is also a challenge. But I think we've got to capture the data first and then find, a simple and compelling way of communicating it.
Yeah, and that's what I want to kind of segue to in our kind of final 10 minutes. Of one of the things is, data, right? Data is what, well, it's generally true in all pharma industry, but in oncology, it's absolutely important. So, we are starting to see, data readouts, not just the immune correlates, not just whether vaccines or OVs, it's not just we're getting, CD8s and or CD4s, it's, we're getting some clinical benefits, whether those are ORR benefits or PFS or OS benefit, whether it's, what Targovax has put out or others. Is there, does anyone want to venture like when we will have like the critical mass, I guess, of data? Will it be one company's kind of key data set? Will it be an approval, like just getting one of these OVs across the finish line? Will it require kind of a number of them? Maybe different approaches to kind of validate the broader concept?
I think all of the above to some degree. I think I want to reiterate what Paul said. We are the oncolytic virus, which are in development right now, are totally different from what was development 10 years ago. So we're basically looking at OV 2.0, if you want to talk about it. And the companies who are developing, of course, have learned the lessons and are much more experienced. So I think we have a much higher chance of success. But I mean, we have to also recognize we're in a sort of a what they call a bear market right now. I mean, the biotech, so any good news not will fall on such fertile ground that it may if you had that same news in 2020. And there was good news. CG Oncology had some good news on the virus. Replimune should come out that could have benefited. So I think it's a string of it. I think not one event alone will be triggered, but yes. More and more OVs having good news, good clinical data will definitely help to change the sentiment. Although having said that, I think that most pharma companies will wait until you have clinical POC before they act. But I think we're very positive here, and we hope that other companies will be successful. That will then definitely show the potential of our technology.
Well, we're at the end of our time, so just quick. Any closing comments from, Lubor kind of had a nice summary in response to my point, but Angelica, Joe, and Paul in kind of that order, kind of quick final summation, questions, comments?
I think we are sort of facing a fantastic future with oncolytic viruses. We are starting to learn how to use them and which patient to use them. So I do think we will see a couple of successful phase 3 trials going forward.
Great. Okay.
I share that excitement. I'm looking forward to what all of you are going to be contributing to that space, and obviously other companies here. I think if we collectively take the learnings from each other, there is really a lot of potential for this modality that I still believe in, and I hope that we all succeed in maximizing that potential.
Paul?
Yeah, I'm very excited too. I think the future's looking bright. I also think there's a potential for companies like ours to collaborate, to work together, to share our technologies and understandings to move us forward even faster. We're collaborating with Targovax on a program, which we're very excited about. So as an example.
Great. Yes. So, well, this has been a great session. I appreciate all of your thoughtful comments and insights. And as a lapsed virologist, as I said on the prep call, I've got a long-standing interest in this space. And so I wish all of you well. Hopefully you all get across the finish line. The more, the merrier. So again, thank you so much.
Thank
you.
Thank you.